Hello. Today I'm going to discuss our work where we assess the feasibility and effectiveness of household pool universal testing with the objective to control COVID-19 epidemics. And this work was published in PLOS Computational Biology, and it was a collaboration between the University of Hasselt, the University of Brussels, the KU Leuven, and the University of Antwerp. By now, we know that COVID-19 is quite an important threat, of course, with respect to public health, as people get sick and they might even die, but also with respect to our economy, as when people are sick, they can't go to work, but also the stringent measures that were imposed to try to control COVID-19 really had a huge impact on our economy. So what best to do to mitigate a COVID-19 epidemic? Of course, try to prevent that people get infected, but that is not so easy. A very effective way is to isolate infected individuals. As logical as it seems, that is very effective. Yet then we of course also need to identify who is infected, and we can do that using PCR testing. PCR testing it's one of the first pathogens where we started doing this to see who is actually infected, and that is very effective to find infected individuals and to isolate them. To better understand how we can use such strategies, how to use PCR testing strategies as efficiently as possible, and we can of course look at epidemiological models, and this is exactly what we're going to do in this study. In this study, we're going to explore the idea of universal testing, and this idea is that we test the whole population in a limited time interval, and we will try to isolate positive individuals. Why would we do that? Because we can, of course, reduce the number of new infections like that. Because we isolate infected individuals, this way they cannot further propagate the virus. And of course, this reduces the number of infections. But additionally, we will also perform a kind of surveillance by testing everybody all the time. We will also get a very good idea how the state of our epidemic is at this time. It's of course a very challenging thing to do because you will need a large number of PCR tests that is required to perform such an action. And therefore, we introduced the idea to use household pooled PCR testing. And that combines the idea of sample pooling, where we increase the number of samples that can be tested with one PCR test. So we combine samples and we use one PCR test to see whether one of these samples is actually positive. And that's what we call sample pooling. To us, it made sense that we combine people that are closely connected, because of course they have the chance that they infect each other, but also if they can come together to the testing area, that makes things a bit more straightforward. So we combine samples of people that belong to the same household. These are added to a pool. And in order to allocate a population of households optimally to a fixed set of pools, we also devised an algorithm. I will not go into the details of this algorithm, but please have a look at our paper where all of this is described in detail. Once we identified individuals, once we identified a positive pool, we will need to isolate the individuals. So there are two ways to do that. Of course, we can take the entire pool and isolate everybody that is in there. That is quite straightforward, yet this also implies that we will isolate individuals that are actually not infected, that are actually negative. And this might really limit the compliance with respect to universal testing, which of course will also impose quite a burden on individuals, as they need to be tested every week, and therefore pool-based testing might induce compliance problems. On the other hand, we can consider individual-based isolation, where we'll find which individual is actually, or which individuals are actually responsible for making the pool positive, and then isolate them specifically. The thing is, we need, of course, additional tests to figure this out. But as the number of pools that test positive is low enough, this can actually be achieved. So these are the two strategies that we investigate in this work. So, as it is a prevention strategy that is really targeted towards individuals, we use an individual-based model. And the individual-based model that we use is Stride, and you can find the model, the source code on GitHub. It's a high-performance C++ individual-based model that can deal with large populations. We'll consider the Belgian population, which is about 11 million individuals. It's a fine-grained model of COVID-19 transmission, and this model has been published recently also in NotCom early this year. And we also have in this model a realistic social network of households, of people can meet each other at the workplace, at the school, and in the community. And in this model, we implemented universal testing to be investigated. And here I show you the results that we obtained. So what you see here is the outcome of the model. And what is shown here on the left, there is a scenario where we will consider universal testing while the epidemic is still going on. And this, what you see here, is the first wave of the epidemic, and at the dotted line we will start universal testing. So when there's still quite a few infections, while there's still quite a few infections that are going on. And we consider different cases. We consider a combination of pool size and the number of tests that are available. So blue is pools of 16 with 25,000 tests, and we consider pool sizes of 16, 32, and number of tests of 25,000 and 50,000. So you can see that the red curve, we have 32 pools of 32 samples and 50,000 tests. So that means that we can test the entire Belgian population in about 1 week. So that is really a short turnaround time. And you can see that even when there are still quite a few infections going on, we can really push down the curve and go to very low numbers in only a few weeks of time. On the right, we have a very similar setting. Again, we have the first wave that is shown here, and we will start the universal testing when the number of cases is down. And you can see that indeed the red scenario keeps the epidemic under control. So that shows us that we can indeed control an epidemic, but interestingly also when we do universal testing when there is still a lockdown going on, so there is stringent measures in place to try to control the epidemic. As you can see here in these box plots, these are different time points. Here we have day 0, and you see 4 box plots. Each of these box plots signifies a particular compliance scenario. I will not go too much into detail here, but you can have a look in the paper. But the trends are pretty clear. At day 1, we still have a lot of infections going on, 1,000 infections that are generated. After day 45, we can see a significant decrease for all compliance scenarios, and at day 90, we actually are converging quickly to 0 cases, actually eradicating COVID, which is of course very interesting. That shows that following this strategy, we could indeed go to 0 cases, even when there is no perfect compliance. So finally, This is already a published paper, so you can have a look. It's published in PLOS Computational Biology, and this was a collaboration with many different people from different backgrounds. So I want to thank all of them for this nice collaboration. So thank you very much, and I look forward to answering any questions that you might have on the Slack channel.